Okay, everybody, we're back again. Now we're going to dive into chapter 9. So this is where all of our prep beforehand, talking about glucose and what glucose kind of does and everything, this all comes to a head where we're going to really dive into glucose metabolism. So we're actually going to pause real quick. There's this little insert that I want to make sure we go back over. So this says, although variability exists in human diets, major portions include carbohydrates, besides fats and proteins. Carbohydrates are one of the major fuels in humans that supply ready and quick sources of energy, mainly in the form of ATP, to other metabolic pathways. In certain situations, carbohydrates, particularly in excess of dietary needs, are converted to fats, and in some other metabolic pathways also are sources of amino acids, the building units of proteins. Cells utilize carbohydrates via oxidation of glucose. Oxidation of glucose. Remember what oxidation is? Oil rig. Oxidation is loss of electrons. Or you can also look at a gaining of an oxygen or losing a hydrogen. Okay. Although other monosaccharides, such as fructose, galactose, and mannose, also come from carbohydrate digestion, all cells use glucose for energy production. Fructose, galactose, and mannose are readily converted to glucose or intermediates of glucose oxidation products and metabolized via glucose oxidation pathway. The overall metabolism of carbohydrates in humans is subdivided as follows. So this is a great little section where all of those definitions of the different glucose-sounding words that we've talked about above, we're going to see here again. So glucose oxidation, glycolysis, so the oxidation of glucose to form pyruvate or lactate. We'll get to this as we dive into the metabolism of glucose. Glycolysis, this is glucose lysis, breaking down the glucose. Conversion of pyruvate to acetyl-CoA enzyme, coenzyme A. Krebs cycle or citric acid cycle or tricarboxylic acid cycle. Metabolism of acetyl-CoA via citric acid cycle enzymes to produce carbon dioxide and reducing agents or electron carriers. And then the respiratory chain or the electron transport chain. We have gluconeogenesis, glycogenesis, and glycogenolysis. And then the pentose phosphate pathway. The next couple of chapters will essentially be looking at each of these different processes and these different pathways and these different things and how they actually happen. Okay? So glycolysis. So here are our objectives. I'm not going to read all of them this time just because there's a lot of them. Take a minute to read through these objectives, highlight a couple of keywords, and be ready. If you need to look at some definitions, amphibolic nature, amphibolic, that's a newer word that we haven't seen before. Go ahead and look that up. Okay, and we'll get into it. Glucose is utilized by all cells. It is used for production of ATP as an energy source, synthesis of glycogen in animals, and starch in plants. Extracellular matrix and cell wall polysaccharides, pentoses, and various other important metabolites. This is what we talked about last chapter. So we already got a lot of that in, exactly what was going on. If you do not recall right now the two starches that we talked about, go ahead and pause and go back up and look at what the names of those two starches were. And then also go back up and look at a couple of those examples of those pentoses, of the polysaccharides. Remember those amylose and the ketose. Take this time, I would even take a minute right now, go back and review those if you haven't since last week. Remember when we talked about in order to convert something to long-term memory, you have to have seen it at least 17 times in a number of different ways. Okay, so take this time to go back and look at that right now. Glucose, so this is glucose going all off into its different directions. So we'll start by talking about glycolysis. So, glycolysis is a metabolic process by which glucose is oxidatively degraded to produce pyruvate aerobically. So, a couple words here. Glycolysis. So, when we're talking about glyco, you can also kind of look at this as glycogen, but think glucose, lysis, breaking down, right? It is a metabolic process. Metabolic process. Now, we've seen metabolic a lot of times. I'm not a robot. Okay. Related to or deriving from the metabolism of a living organism. I want to see better. Metabolic, related to or deriving from the metabolism of a it kind of definition goes back and forth. But here's where we go. The chemical changes that take place in a cell and organism. That's what we want. Sometimes the definitions will go back on itself, but we just want to find a good one. Okay? Specifically, these changes make energy and the materials cells and organisms need to grow, reproduce, and stay healthy. That is what we mean by a metabolic process. Okay? Now, glucose, that's our main focus, is oxidatively degraded. So take a minute, think about what that means. Oxidatively, remember, oxidation is loss of electrons. So we're going to think losing those electrons, things are going to be being pulled away. In order to produce pyruvate, we'll get into exactly what pyruvate is. Pyruvate's another chemical. We'll talk about why it's important and kind of its function and its source. Aerobically. Okay, so this is really important to understand. Aerobic versus anaerobic. This, I can tell you right now, this is probably one of the most missed things on tests. Just these two different words. So aerobic means with air and refers to the body producing energy with the use of oxygen. And we'll get more into this later on. So we have use of oxygen to produce pyruvate. Now, a little bit earlier, we talked about something also called lactic acid, right? Or lactate. Lactate is going to be the result of glycolysis in an anaerobic environment. So no oxygen is present. Okay? Super, super important thing to understand. When there is oxygen present, we get pyruvate from glycolysis. When there's no oxygen, we end up with lactate or lactic acid. We'll get a lot more into this later on. So right now, let's just focus on the words that are up here. Okay, so glycolysis occurs in the cytosol. So we are inside of the cell. Remember, we have our phospholipid bilayer, we have our nucleus, and then everything that's all through here, all of the jelly-like stuff, is our cytosol. So that's where glycolysis is going to occur. We have 10 enzymes catalyze the glycolytic pathway. 10 enzymes. Okay, so this is going to be where I talked about before, where we need to focus on the enzymes, knowing and understanding what those enzymes do. A lot of your test questions are going to come from that specifically. Knowing what the enzymes are and what they do, where they're located between steps, as well as which of them are the rate limiting step. Okay? So the energy released during the conversion of glucose to pyruvate is conserved in the form of ATP and the reduced coenzyme NADH. So if we think glucose gets oxidized, so we have a reducing agent, this time coenzyme, that NAD plus gets reduced to NADH. Glucose transport into cytosol. So glucose diffuses through the membrane via glucose transporters into the cytosol. Insulin stimulates the glucose transport into the muscle and the adipose tissue by moving glucose transporter proteins. Remember those GLUT? It moves the GLUT transporter proteins to the cell membrane. Specifically, we mentioned GLUT2. Don't worry too much about all that right now. Just understand that it's a GLUT transporter protein. Later on, we'll learn all about, there's GLUT1, GLUT2, GLUT3, and GLUT4. Okay, but we'll talk about those more in physiology 2 down the road. So GLUT deficiency leads to diabetic condition. Remember what we talked about when there's too much insulin? There's a whole bunch of insulin there, and these get overstimulated. They get stimulated over and over and over again. It can cause them to down-regulate. In other words, kind of absorb back through the membrane so that there's less of them. And that's what the cause of type 2 diabetes is, that deconditioning or overconditioning to the insulin. Okay. Diving back again into glycolysis, we're going to look at 2 phases. There's a priming phase, also known as the preparatory phase. And this is going to use 2 molecules of ATP to activate glucose in the initial steps of the glycolytic pathway. If you've ever heard of the expression, you have to spend money to make money, it's that kind of concept. You have to use some energy to create more energy. So we're going to use 2 molecules of ATP to activate the glucose. And then in the payoff phase, we'll actually produce 4 molecules of ATP and pyruvate. So if we think in total in glycolysis, we make 4 ATP, but we net 2 ATP. This will be a tricky wording on questions, where he'll ask you how much net ATP do you make, or how much ATP do you make, how much ATP is made in the phase of glycolysis. 4 is made. But how much do we net ATP? How much ATP do we net? 2. 2 different questions, very similar the way that you're going to ask them. Look for that keyword net of ATP. Because if the question doesn't clarify, and you have both the answers 4 and 2 and the question does not clarify, I would lean more towards, I think most often they want the net version, but you can always go and argue that question and you should get points back for it, because you need to know, is it how much it's made, or how much is net ATP. And here is our beginning steps to glycolysis, okay? This is the preparatory phase. So what we want to know from here, first and foremost, is the reactant, the product, and these enzymes, okay? So we want to know all of this stuff. This is something I highly recommend that you take time, and you can do a couple of different things. Either you can print out note card, or make little flashcards with all of these, with all of the names of everything, and you can literally put them on the table and arrange them in the correct order and matching up where the different enzymes go. Or I would also highly recommend writing this process out, probably like 10 or 15 times. Then write it out 2 or 3 times, looking at it, and then start to try to write it out at least once a day from memory. And if you can do that and then just keep working on it, come test time, if you can just immediately, as soon as you start your test, write this down or have a mental map, mental picture of it, that will help you so much for your exam. Okay, especially here with these enzymes. You have to know the enzymes really, really well. I don't know why 5 is on here. Okay. And even same thing here. Know the products and the reactants. You need to know the names all of here. And you also need to know any of the steps that makes NADH, makes ATP, or uses ATP, or uses ATP. Okay, you need to have those on. Honestly, this is the set chunk where a lot of people seem to really, really struggle because they don't spend the time to learn this whole process. This takes time to learn, and there's really no other option besides just grinding it out and kind of writing it over and over and over again. You can abbreviate a lot of these things. So like glucose, you can write G6-phosphate or G6P, F6P, F16BP. This one I would write either GA3P or something like that, something along those lines. DHA, DHAP, etc., etc. Find ways to abbreviate it, so that way if you're on the test, you can still look at answer choices and know, okay, that's what whatever stands for, that's what it means. Okay. I'm not going to go over a lot of this right now. This is something I really want you guys to go over on your own. Just go over it and over it and over it and over it. One thing that I will point out really quick. 1 molecule of glucose goes in, and 2 molecules of pyruvate come out. Okay? Glucose is a 6-carbon molecule. Right? 1, 2, 3, 4, 5, 6. 6 carbons. Pyruvate, each pyruvate is a 3-carbon molecule. Remember that. It's a very important thing to remember, that you get 2 pyruvate for each glucose that you produce. He'll ask questions. They'll ask a lot of questions about stuff like that, especially later on when you add the next step after pyruvate, where the pyruvate goes to make more ATP. So you want to remember that each glucose makes 2 pyruvate. Okay, this chart right here talks about the 3 possible fates of the pyruvate formed in glycolysis. Know this chart like the back of your hand as well. There will be 2 or 3 test questions that come directly from this, mostly focusing on this right here, where you have 2 pyruvate. In anaerobic conditions, what do you get? Lactate. In aerobic conditions, it continues to acetyl-CoA into the Krebs cycle. In hypoxic or anaerobic conditions with yeast specifically, it creates ethanol. This is the process of making alcohol. It's like this is the process. Actually, this is what yeast goes through to make beer, to make wine, all that sort of stuff. The following reactions of glycolysis are shown as single reaction for convenience, and also to specify the enzymes with the required cofactors. Note here all the carbohydrate molecules in human metabolism are D isomers. For simplicity and some illustration, we are not writing it. Okay, now we're going to go step by step through the entire process of glycolysis. Okay, and that's this whole chapter. Step 1. we have our glucose, a 6-carbon molecule, gets converted to glucose 6-phosphate. Now when we add a phosphate, adding a phosphate is what type of enzyme? The kinase. So remember, kinase. This reaction is catalyzed by hexokinase. You can think of it, glucose is in the shape of a hexagon, kinase, it phosphorylates it. It requires magnesium ion as its cofactor. Little note here, all kinase enzymes require magnesium ion as their cofactor. Please write that down. This is very, very helpful information. Every single time that you see a kinase, know that you need magnesium. If you can just remember that one thing, that'll make everything way easier. Put this into a chart or something with your enzymes. Requiring magnesium ion as its cofactor, which complexes with the second substrate ATP. Complexes just means that they join up. The second substrate ATP. So we take a phosphate from the ATP. Our magnesium helps facilitate the movement with hexokinase to get our glucose 6-phosphate, or you can call it G6P. This is the first ATP requiring priming step. In all tissues, several hexoses can be phosphorylated at carbon 6 by this enzyme and its isoenzymes. However, in liver tissue, the substrate for this hexokinase is only D-glucose, and it is known as glucokinase. So this is the exception, right? So whenever we have exceptions, we want to write those down. So in the liver, it's known as glucokinase and not hexokinase, which is, it's technically hexokinase 4, but we'll call it glucokinase. So write that down. Liver equals glucokinase. Everywhere else, hexokinase. Also note here, this reaction is an irreversible reaction and highly exergonic. Irreversible reaction and highly exergonic. Formation of glucose 6-phosphate is a critical and important step, as it is in the junction of several other important metabolic pathways, such as glycolysis, gluconeogenesis, glycogenesis, glycogenolysis, and pentose phosphate pathway. Again, if you don't remember what all of these words mean, except for we haven't talked about pentose phosphate pathway, if you don't know how to define each of these words in your own words, please take a minute and do so. There we go. Hexokinase is present in all tissues. Glucokinase is present in the liver. There are 3 major purposes to synthesize glucose 6-phosphate, including other phosphorylated intermediates in glycolysis are phosphorylated intermediates, including glucose 6-phosphate, once formed, cannot leave the cell because they don't have specific transporters for phosphorylated sugar molecules, and also they are charged molecules, and plasma membrane does not allow them to cross due to a barrier. This we mentioned a while back, that once glucose enters and is phosphorylated, it is not able to leave. Once it is phosphorylated, it cannot leave. So this is specifically, we're going to be talking about all of the tissues except for the liver. The liver is again the exception to a lot of rules. Phosphorylated intermediates are the essential substrates for the glycolytic enzymes and useful source of phosphoryl group containing energy molecules required for the enzymatic steps. binding energy obtained from the binding of the phosphate groups to the active sites of enzymes decreases the activation energy and increases enzyme specificity for these substrates and the reactions. Pause for a sec and reread through that and make sure you understand everything that that's saying. In particular, the phosphate groups of ADP, ATP, and other phosphorylated intermediates of the glycolytic pathway form complexes with magnesium ion, and most glycolytic enzymes use magnesium ion as a required cofactor. So that's a good thing. So most glycolytic enzymes use magnesium ion as a required cofactor. So if you have to guess, if you don't remember, guess magnesium. This is also something that's interesting because a lot of people, because we don't eat enough green leafy vegetables and just vegetables and fruits in general, most of the population is actually low on magnesium. It's a really, really good supplement to give to a lot of people, especially if they're feeling low energy, if they're having dietary issues, if they're having low energy, they're feeling run down, depression, that sort of stuff as well. Magnesium seems to be a really good way to help those kind of patients and those kind of people. Next step. Step 2 in the isomerization of G6P to FR6P. Now, isomerization, remember what an isomer is. The exact same chemical makeup, just rearranged. That's what an isomer is, isomerization. When we talk about our enzymes, we should recognize this isomerase. Now, if we look at the name here, phosphohexose isomerase. We just looked at hexo kinase, so we phosphorylated the hexo. And now it is phosphorylated hexose. Phosphorylated hexose, that ose means sugar. Phosphorylated hexose, isomerization. See how the enzyme tells us exactly what it's doing? So it's taking that glucose 6-phosphate and turning it into fructose 6-phosphate. So in the second enzyme catalyzed reaction, glucose 6-phosphate in aldose is isomerized to fructose 6-phosphate, a ketose. So it goes from an aldose to a ketose by the enzyme phosphofructoisomerase, requiring magnesium ion as its cofactor. This reaction is a reversible reaction. This is another thing he'll like to ask. He'll like to ask which ones are reversible or which ones are irreversible. Me personally, I would pick one or the other, whichever has less. Know which of all of them are irreversible and memorize those. And then I know that all of the rest are the other choice. Formation of fructose 6-phosphate is thermodynamically favorable, but it can be reversed too because there's relatively a small change in free energy. Also note here, by us isomerizing glucose 6-phosphate to fructose 6-phosphate, it is actually favoring the addition of the second phosphate group in the next reaction at carbon 1, and thereby balancing the structure to be degraded from the middle of the molecule, as well powering the molecule with the attachment of the second phosphate group by making it energetically ready for the next step reaction. Okay, so we know that next steps we're going to see are another more priming, and we're going to use another ATP. So it's prepping it to be able to accept another phosphate. Because remember, our glucose right now has 6 carbons. We eventually want to split it into 2 to have 2 3-carbon pyruvates. Second phosphorylation, 2'. This one right here, PFK1, phosphofructokinase 1. Look at that. Phosphorylated, fructose, kinase. Kinase is going to phosphorylate stuff. So our phosphorylated fructose gets phosphorylated again. See how it says exactly what it's doing. And it'll also give you a clue what it is before and after. Okay? We have fructose 6-phosphate, a single phosphate. And now we have fructose 1,6-bisphosphate. Bi meaning 2, so it's phosphorylated twice, and it's at the 1 position and the 6 position. So you can kind of see how logically this progresses. So in the third step, phosphorylation of fructose 6-phosphate occurs at the first carbon producing fructose 1,6-bisphosphate, catalyzed by PFK1, requiring magnesium ion as cofactor, which is complexed with the second substrate ATP. This is the second ATP requiring priming phase reaction. This reaction is also an irreversible and highly exergonic reaction. This step is known as the committed step in glycolysis. Now the cleavage. So we look at this. So remember what we talked about a little bit ago? We said that the isomerization, aldose to ketose. We have fructose 1,6-phosphate, which is a 6 carbon, is further catalyzed and splitted to half by fructose 1,6-bisphosphate aldolase, or simply aldolase, a lyase class of enzyme. So our aldolase, which is a aldose is a type of sugar. Got our sugar, the lase breaks it apart, so aldolase. And if it helps you to remember the whole name, fructose 1,6-bisphosphate aldolase, remember it that way, because then it's telling you exactly what it is that it's splitting. To form 2 3-carbon phosphorylated compounds, dihydroxyacetone phosphate and a glyceraldehyde 3-phosphate. Now this reaction is reversible. In humans, this enzyme does not need any bivalent cofactor, whereas in bacteria and in plants, zinc ion is used as a cofactor. So this step does not require a cofactor in humans. I do recall him asking either a question on the quiz or on the exam referring to zinc in this one in bacteria or... Alexa, stop. I do remember him having a question about zinc somewhere in there. Step 5, isomerization. Triose phosphate isomerase catalyzes the conversion of the ketose, dihydroxyacetone phosphate, to an aldose, glyceraldehyde 3-phosphate, without requiring any cofactor. So we had a dihydroxyacetone phosphate and a glyceraldehyde 3-phosphate. This is the one that we want. So we're going to convert this. We're going to isomerase, convert it to another glyceraldehyde 3-phosphate. So now we started with that 6-carbon glucose, and now its oxidation up to this point yields 2 molecules of 3-carbon triose phosphates. In this reaction, 2 molecules of glyceraldehyde 3-phosphate is produced. So now we have 2 molecules of glyceraldehyde 3-phosphate. Okay, from here on out, I'm not going to keep reading all of these. Oh, actually, no, we're almost done, so it'll be fine. Step 6. Oxidation of aldehyde to acid. From this point, glycolysis enters into the payoff phase. Know this cutoff. Know that the cutoff is when it gets split into two 3 carbons, and then you end up with 2 glyceraldehyde 3-phosphates. So now we invested all of that ATP, those 2 ATPs, so now it's time to start reaping the rewards. So we're going to start to produce ATP as well as NADH as electron carriers. So glyceraldehyde 3-phosphate reacts with inorganic phosphate in the presence of oxidizing agent as well as the coenzyme NAD+. Can you think back? Can you recall what B vitamin is involved with that? niacin. Do you remember the mnemonic? Tall, rich, nudists played pickleball for centuries. We have B1, B2, B3. So NAD plus is our B3. And forms 1,3-bisphosphoglycerate catalyzed by glyceraldehyde 3-phosphate dehydrogenase. And also produces NADH as a carrier of electrons. One molecule of glyceraldehyde 3-phosphate produces one molecule of NADH. So because we have two of them, we get 2 NADH. Now each NADH, here's a number that we'll want to remember. 1 NADH can produce 2.5 ATP. This will be discussed later on in the electron transport chain, how that actually occurs, but keep that number in your head. So this NADH is created but is not used in this pathway. It instead gets used later on. Now phosphoryl transfer, substrate level phosphorylation. 1,3-bisphosphoglycerate now reacts with ADP, catalyzed by phosphoglycerate kinase, requiring that magnesium ion as a cofactor, and forms 3-phosphoglycerate and generates the first molecule of ATP. One phosphate group is transferred to ADP to form ATP, noting the rate of the forward reaction is higher than the rate of the backward reaction, ensuring formation of ATP. So this is technically a reversible reaction, and it can go back and forth, but the forward reaction rate is higher, so it's more likely to end creating the ATP. Step 6 and Step 7 reactions are coupled, and the overall reaction is exergonic. Although not irreversible, but reversible in cellular conditions, this step is the first substrate level phosphorylation that generates ATP in glycolysis. This is called substrate level phosphorylation to distinguish it from the respiratory chain relation ATP. Remember this, substrate level phosphorylation. Know that when you're talking about substrate level phosphorylation, you're talking about glycolysis, and you're talking specifically about this type of reaction, where we have the dephosphorylation phosphoglycerate kinase, taking it from the 1,3-bisphosphoglycerate, adding it to the ADP to create ATP. Remember, so in this step, we create 2 ATP total because we have 2 1,3-bisphosphoglycerate. Okay. Mutation of 3-phosphoglycerate to 2-phosphoglycerate is catalyzed by a phosphoglycerate mutase, which transfers the phosphate group from 3C to 2C. This uses magnesium ion as a cofactor. It is important to note the 2,3-biphosphoglycerate is an intermediate during this conversion and plays a major role by activating the catalytic cycle of this enzyme. We talked about mutase. Mutase is similar to an isomerase, but a little bit different, where it just moves that one individual thing. So phosphoglycerate mutase, it mutates, changes one thing on that phosphoglycerate. It changes it from being bound to the third carbon to being bound to the second carbon instead, to prepare it for the next step. Finally, step 9, dehydration of 2-phosphoglycerate to the unstable phosphoenolpyruvate is catalyzed by enolase enzyme requiring magnesium or manganese ion. 2-phosphoglycerate to a phosphoenolpyruvate. Come back up here real quick. Phosphoenolpyruvate. So technically, this is kind of where those reactions end, but we count it as one more step of those being broken down, degrading further into pyruvate and creating that second ATP. The second ATP per glyceraldehyde 3-phosphate that we made earlier. So 1, 2, 3, 4 4 ATP, and 2 NADHs. So if we think about the total potential amount of ATP created here, there's minus 1, minus 2, plus, it puts it back to minus 1, back at, actually this puts us back at 0, at positive 2. Each of these is worth 2.5, so technically glycolysis, as long as we have oxygen and the electron transport chain is running, glycolysis will actually produce a net total of 2, 4, 9 ATP, as long as there's oxygen, and this can go to the electron transport chain. Or if it's in anaerobic conditions, we'll have creation of 4 ATP. Net 4 ATP total, 2 ATP net. Okay, so like I said, take your time, draw this out a couple of times, write this out. You don't have to write all of the drawings and everything that's going on, but start getting used to recognizing the names. It also really helps to practice the pronunciation of these names. If you can pronounce them clearly and accurately, you're much more likely to be able to write them as well as remember them and understand them and what they do. Okay? And next time, we'll get into the Krebs cycle, which is a whole other beast. So good luck with all this material. Good luck on all of your studying, and good luck on any future exams you have soon. Thank you so much for watching. I'll see you guys later.